what is life?
Mm.
The physical aspect of the living cell. Erwin Schrödinger.
Mm.
Imagine looking at this microscopic speck of matter, right?
Hmm.
And it's just completely battered by the violent, random thermal chaos of the universe.
Oh, yeah, just absolute molecular
Right,
chaos.
but then you realize it holds this unbroken memory that spans, literally a billion years.
It's incredible to think about.
And every single cell in the person listening to this deep dive right now is doing exactly that.
Yeah.
So today we're really diving into one of the most heavily debated questions in the history of science, which is, can the stability, the inheritance, the organization, and the apparent order of living systems actually be explained through just physics and chemistry?
Right, and it's a massive inquiry. I mean, we're looking at how a physicist, approached the ultimate biological mystery.
Exactly. And to figure out which of his influential proposals remain scientifically meaningful in this age of modern molecular genetics, we're going to trace a very specific path. So for the next stretch of this deep dive, we'll investigate the puzzle of hereditary stability. Like, how do traits survive that chaos?
Yeah, that's the big one.
And we'll decode Schrödinger's famous proposal of the aperiodic solid. We really need to look at how molecular information is actually stored and the physical reality of genetic mutation. Right?
Absolutely.
And how biological order is maintained against the relentless pull of entropy. So we'll break down free energy, map that against our modern understanding of DNA in the cellular context, and see how evolution balances reliability with, necessary randomness.
Right.
And finally, we will weigh the historical influence of the book alongside its pretty hard limitations.
Yeah, and to do that fairly, I mean, we really have to ground ourselves in a very specific temporal context.
Okay, set the stage for us.
So Schrödinger delivered the lectures that actually formed this book in Dublin, and this was in 1943. Now, at that point, classical genetics had already established that chromosomes existed, right? And that genes were central to inheritance. But the actual chemical identity of the gene was, it was entirely unsettled.
Right. They knew there was some physical vehicle for heredity, but scientists at the time, a lot of them just assumed it had to be proteins.
Yes,
Yeah,
exactly.
because proteins possess the chemical complexity that they thought was required for life. I mean, DNA was known, but it was kind of widely dismissed as this, stupid, repetitive structural molecule.
Yeah, they thought it was just scaffolding.
Precisely.
So because of that, we need to be incredibly rigorous with our language today.
Okay, how so?
Well, we have to clearly distinguish between what Schrödinger proposed theoretically in the 40s, what the historical evidence indicated at that exact moment, and then what modern biology explains with current data.
Right.
And also what just remains unresolved. I mean, we cannot project modern systems biology backward onto his work.
That makes total sense. And I think that brings us to the first major conceptual hurdle here. If we want to understand life physically, we have to recognize why the physical scale of inheritance was just this absolute paradox to a physicist in the 1940s.
Oh, it was a massive headache for them.
Right, because the classical physics relies on bulk laws.
Hmm.
So if you think about the thermodynamic stability of a room, its temperature, its pressure,
Yeah.
that's essentially a statistical illusion.
It is, yeah.
It's an average derived from, trillions and trillions of gas molecules just colliding in absolute microscopic chaos.
Exactly. It's the law of large numbers. Basically, statistical mechanics dictates that the larger the population of particles you have, the smoother and more predictable the macroscopic behavior is going to be.
Okay, but a gene violates that premise completely.
Completely.
Because a gene is a microscopic cluster of molecules. It doesn't have the luxury of trillions of particles to just average out the noise.
No, it's incredibly tiny.
So the friction here is really obvious. How does a tiny nanometer-scale cluster of atoms reliably transmit complex morphological traits, like the exact shape of a wing or, a retina, across thousands of generations without being completely scrambled by the ambient thermal energy of the organism?
Right, because if heredity relied on those continuous statistical laws of macroscopic physics, the genetic information would degrade in seconds. It would just melt away.
Just from the heat of the body.
Exactly. So Schrödinger turned to the microscopic laws, which is quantum mechanics.
Okay.
He proposed that the incredible stability of the gene is fundamentally a quantum mechanical phenomenon.
How does that work?
Well, in the classical view, energies are continuous, right? Things can slide gradually from one state to another, like a ramp.
Right, okay.
But in the quantum realm, molecules exist in discrete, highly stable configurations that are separated by immense energy barriers.
Meaning they don't just slowly melt into a new shape.
No, they can't.
The covalent bonds that hold a molecule together require a very specific, discrete quantum of energy to actually break or alter.
Yes.
They're trapped in these steep energy wells.
Exactly. To transition from one stable molecular configuration to another, it requires a discrete jump.
Like a physical leap.
Yeah, an isomeric transition to overcome that barrier. And this is why atoms don't just randomly morph into a disorganized puddle at regular body temperature.
Wow, okay.
They are locked into these long-lived structures by the strict quantum mechanical rules of electron sharing.
Okay, but I think we really need to pause here and address a major misconception. This is misconception number 4 on our list.
Oh, the quantum mysticism one.
Yes, because when we bring quantum mechanics and life into the same orbit, it is so easy to slip into that quantum mysticism space.
Very easy, yeah.
This idea that life operates on some spooky non-local quantum life force, or that, DNA is tapping into quantum fields to process information or something.
That is such a crucial correction. I mean, we must absolutely reject that reading of the text. Schrödinger was not invoking any mysterious, vitalistic quantum force. Not at all.
Okay, so what was he saying?
He was merely pointing out that standard foundational chemistry is governed by quantum mechanics.
Just regular chemistry?
Exactly. Quantum mechanics explains the structural integrity of covalent bonds, and that explains why a molecular structure can be stable in the first place.
Ah, I see.
And it also explains why a mutation, which is just a chemical alteration to that structure, has to be a discrete physical jump. It's not a gradual slide.
Right, so he's really just explaining the physical strength of the building materials. He's not claiming the building has a ghost in it.
That's a great way to put it. We do not need exotic quantum principles to explain biological mutation.
Okay.
The mechanics of mutation are fully explained by chemical kinetics, replication damage, and just the physical dynamics of chromosomes in a cell.
So quantum mechanics is really just the basement foundation that the entire house of chemistry and therefore biochemistry is built on.
Correct. It's just the ground floor rules of reality.
Okay, but that stability creates a new problem then. Because if heredity requires this incredibly stable molecular structure locked in deep energy wells, how does that structure actually hold the vast complexity needed to build an organism?
Right.
Because stability usually implies repetition, right?
Yeah.
Like a crystal.
Yeah.
And repetition doesn't carry complex information.
And this is exactly where Schrödinger introduced one of his most enduring theoretical concepts. He called it the aperiodic solid.
The aperiodic solid is such a brilliant physical reconciliation when you think about it.
It really is.
Because if you look at a standard periodic crystal, say like a diamond or a lattice of table salt, it is incredibly stable because the atomic arrangement repeats identically in all directions.
Over and over.
But that strict periodicity means its information capacity is practically zero.
Right.
It's like typing the word "the" over and over for 1,000 pages.
Exactly.
On the flip side, you have a gas or a liquid, which is incredibly complex and variable, but it has no structural memory. It just dissipates.
It just flows away, yeah.
So to get both structural stability and high information density, you need a solid that doesn't repeat.
Yeah.
And a periodic solid.
And when we test that theoretical deduction against modern biology, the alignment is just striking.
Yeah.
I mean, the DNA polymer maps beautifully onto this concept.
How so?
Well, the structural backbone of the DNA double helix, the alternating sugar and phosphate groups, that part is highly regular, repetitive, and thermodynamically stable.
Okay, so that's the solid part.
Right, that fulfills the solid or crystalline requirement.
Yeah.
But the sequence of the nucleotide bases that are tucked inside that backbone, adenine, thymine, cytosine, and guanine, that varies continuously without a strict repeating pattern.
So that varying sequence is the aperiodic component.
Exactly.
And that's what allows a single microscopic molecule to store an astronomical amount of biological information.
Precisely. However, this brings us to misconception number 1.
Okay.
And it is a massive historical error that we really have to correct here.
Let's do it.
Because of how perfectly the aperiodic solid maps onto DNA, there is this widespread belief that Schrödinger discovered or precisely predicted DNA, the double helix, or complementary base pairing.
Right, which completely ignores the actual history of molecular biology.
Completely.
Because Schrödinger outlined a theoretical physical boundary condition for what the hereditary material had to achieve.
Yeah.
But deducing a physical requirement is not at all the same as identifying the actual chemical molecule.
Exactly. I mean, molecular genetics wasn't built by theoretical physicists musing about quantum states in armchairs.
No.
It was built by rigorous, often grueling experimental work by cytologists, structural biologists, and biochemists in the lab.
Right. The real evidentiary proof that DNA was the carrier of genetic information came from Avery, McLeod, and McCarty in 1944.
Yes, exactly.
And then the physical architecture of the double helix was subsequently solved by Watson, Crick, and Rosalind Franklin through X-ray crystallography and, empirical model building. So Schrödinger provided this intellectually stimulating framing, but he did not discover the DNA molecule.
He absolutely did not. And honestly, if we push the crystal analogy too far, it actually distorts how DNA works.
Yeah, because calling it a solid or a crystal implies rigidity.
Right, like it's a frozen rock.
But inside the nucleus of a living cell, DNA is violently dynamic.
Oh, it's chaotic.
It's constantly bending, it's unzipping for replication, it's melting, supercoiling around histones, it's being chemically tagged with methyl groups. It exists in this crowded, highly reactive aqueous environment.
Yeah, it's swimming in water and proteins.
So if DNA were actually a rigid crystal, the cell couldn't read it. The information would just be locked away forever.
And that is the exact limitation of the metaphor. It captures the stability of the linear sequence perfectly, but it completely fails to capture the immense physical flexibility and chemical reactivity required for the molecule to actually function within a living system.
Right. Which naturally leads us to how that information is actually used.
Yeah.
Because Schrödinger referred to this aperiodic sequence as a code script.
Yes, he did.
And in the modern era, our brains instantly map that onto computer science.
Oh, immediately.
We think of the genome as software, like a deterministic program running on the cellular hardware to compile an organism.
It is the dominant metaphor in popular science today.
Yeah.
And it is fundamentally flawed if you take it literally.
Right, because it feeds directly into misconception number 3 on our list.
The blueprint idea.
Exactly. The idea that genes contain a complete deterministic plan for an organism, like a blueprint for a house.
And the blueprint analogy fails on almost every level of biological reality.
Really? Break that down for us.
Well, just to clarify our terms first. When modern biology refers to the genetic code, it means something very specific and restricted.
Okay.
It refers to the chemical dictionary. The exact mapping of nucleotide triplets in RNA into specific amino acids to build proteins. It is a translation mechanism. It is not an architectural master plan for the whole organism.
That difference is huge. I mean, a cell is not a generic silicon chip passively executing a detached string of code.
No, not at all.
In a laptop, the hardware doesn't change based on whether you're running a word processor or a video game.
Right, the circuits stay the same.
But in a cell, the hardware, meaning the proteins, the transcription factors, the metabolic networks, they are constantly reading, repressing, and physically altering the expression of the software.
Yes.
And the DNA, in turn, is producing the very hardware that regulates it. They're in this state of constant reciprocal causation.
It's a feedback loop. And this completely changes our understanding of heredity.
Yeah.
You don't just inherit a code script from your parents.
What do you inherit?
You inherit what modern biologists call distributed heredity.
Distributed heredity, which is why, if you take a completely synthesized human genome and just inject it into an empty lipid vesicle, nothing happens.
Exactly.
You don't get a human, you just get inert chemistry sitting there.
Right. When a zygote is formed, it certainly inherits nuclear DNA, and that DNA is the essential, stable memory bank of sequence information.
Okay.
But that zygote also inherits maternal RNA, functional ribosomes, fully formed membrane structures, mitochondria with their own separate DNA, incredibly specific chemical gradients, and complex epigenetic states.
It's inheriting a whole functioning factory, not just the manual.
Exactly. The DNA provides the sequences for the proteins, but the pre-existing organized cellular system is completely required to interpret that sequence.
That makes a lot of sense.
Because embryonic development is a highly self-organizing process. It's driven by gene regulation, metabolism, mechanical tension in the tissues, and environmental conditions.
Wow.
It is not just a blind readout of a linear script. The script only has meaning within the specific context of the interpreter.
That's a great way to put it. But maintaining that interpreter, maintaining the staggering complexity of that cellular hardware, constantly proofreading the DNA, actively transporting molecules around the cell, that requires work.
Oh, an immense amount of work.
Serious, continuous physical work. And in physics, Work brings us face to face with the second law of thermodynamics,
Yes.
the universal tendency toward disorder and entropy.
The grim reaper of classical physics, basically.
Right. Every physical system degrades eventually. Heat dissipates, gradients flatten, structures collapse. So the fundamental paradox of the living cell is how an organism maintains this exquisite, highly ordered biological state without violating the laws of thermodynamics.
It seems impossible at first glance.
And Schrödinger's phrasing for this became incredibly famous. He argued that life feeds on negative entropy.
And that phrase, while it's highly evocative and it was hugely influential, it has spawned decades of confusion.
Yeah.
It leads us squarely into misconception number 2.
Which is?
The belief that living organisms somehow violate, circumvent, or reverse the second law of thermodynamics.
Which they absolutely do not.
No.
Because when you look at a tiny seed growing into this massive, highly ordered oak tree, it visually looks like entropy running in reverse.
It does, yeah.
But that's only if you draw your boundaries incorrectly. The second law states that the total entropy of an isolated system always increases. But no living organism is an isolated system.
Exactly. We are open systems.
Right.
We exist in a constant state of non-equilibrium. We continuously exchange matter and energy with our environment.
So modern biology has largely discarded the term negative entropy
Yes.
in favor of a much more rigorous thermodynamic concept, which is free energy.
Right. Free energy. Organisms survive by constantly capturing free energy from their environment.
Like eating.
Exactly. Plants strip it from the electromagnetic radiation of the sun. Animals extract it from the chemical potential energy locked in the bonds of the food we eat.
And the mechanism by which the cell uses this free energy is fascinating.
Yeah.
It relies on metabolic coupling.
Yeah, metabolic coupling.
Right. And to explain this without relying on, high school textbook cliches,
Yeah.
we have to look at it mechanistically. The cell needs to drive energetically unfavorable reactions, like forcing ions against a concentration gradient, or stitching amino acids together into a complex protein.
Things that take effort.
Right, these things will not happen spontaneously. They require a direct input of energy.
So the cell couples that unfavorable reaction to a highly favorable energy-releasing reaction, typically the hydrolysis of a molecule called ATP.
It's essentially a microscopic mechanical gear system.
Yeah, that's a good visual.
The violent release of energy from breaking a phosphate bond on ATP
Right.
physically forces a protein to change its conformation.
Right.
It's like cocking the hammer of a gun or turning a crank. You're using the chemical currency of free energy to physically force the biological machinery to do mechanical work that it otherwise mathematically just couldn't do.
But, and this is the crucial thermodynamic trade-off we have to mention,
Okay.
that coupling process is far from 100% efficient. To maintain its internal local order, the organism must release heat into its surroundings. Furthermore, it takes in high-energy complex molecules as food and exports lower-energy, highly disordered molecules as waste.
Which means if you draw the boundary of your system to include the organism and its surrounding environment, the total entropy production of that combined system is still relentlessly increasing.
Always.
Like every listener to this deep dive right now is radiating around 100 watts of heat into their room and exhaling degraded carbon dioxide, accelerating the universe's overall entropy just to keep their internal cellular structure intact.
Yeah, it's a constant drain.
We don't defy the second law. We surf it.
We surf it. I like that.
Life operates strictly through thermodynamic flows.
Exactly.
So we have this highly stable molecular memory in DNA, and we have an open thermodynamic system burning free energy to maintain the hardware that reads it.
Right.
But this combination creates a profound evolutionary friction.
How?
Well, if the system is perfectly stable and perfectly maintained, life is static. A perfectly replicated sequence never changes, which means it can never adapt.
Ah, I see.
So how do we get the reliable stability required for heredity coexisting with the variation required for evolution?
Well, Schrödinger addressed this by distinguishing biological order from the statistical order of bulk physics.
Okay.
He called the biological mechanism order from order.
Order from order.
Right. The idea that a cell reproduces accurately because its existing highly specific molecular organization directly templates the new organization.
Which sounds elegant,
So?
But when we look at modern cell biology, the interior of a cell doesn't look like a pristine clockwork templating machine.
Not at all.
It is unbelievably chaotic.
The modern reality is stochastic noise. A cell is a profoundly noisy environment.
Why is that?
Because we are dealing with nanometer scales and often very low copy numbers of specific molecules, thermal energy causes constant random collisions. Transcription factors don't just glide smoothly to their targets.
Right.
They bounce around randomly until they accidentally collide with the correct binding site on the DNA. Gene expression frequently occurs in sporadic, unpredictable bursts.
And the DNA itself is constantly under attack, right?
Yeah.
The thermal noise of the body, metabolic byproducts, background radiation.
Oh, yeah, constantly.
I mean, every second, the DNA in our cells is suffering thousands of microlesions and physical alterations. We are only surviving this conversation because our cellular hardware is frantically compensating for it.
It's working overtime, which reveals the true brilliance of biological evolution, honestly. The organization of the cell doesn't somehow freeze out this thermodynamic noise. It actively manages it.
Right.
Biological reliability and molecular randomness exist simultaneously.
So we have these layered mechanisms of dynamic robustness.
Exactly.
Molecular proofreading enzymes that ride along the DNA during replication, physically feeling for the wrong geometry of a mismatched base pair and excising it.
Yeah, they literally cut it out.
We have complex repair pathways constantly surveying the genome for damage. We have immense redundancy built into genetic regulatory networks, so if one pathway fails due to a random fluctuation, another can just buffer the shock.
Right. So the order we see in biology isn't the perfect, rigid order of a crystal. It is a resilient, dynamic equilibrium maintained at great energetic cost.
But crucially, the cell doesn't fix everything.
No, can't.
If the repair mechanisms were 100% perfect, evolution would just grind to a halt.
Completely.
Noise isn't just an error in the system. It is the generator of biological novelty.
Yeah.
Those random molecular events, the occasional copying errors that slip past the proofreaders, the unrepaired chemical damage, those are the physical reality of mutations.
They provide the raw, unguided variation. And here we see how the physical requirements and the biological requirements interlock perfectly.
How do you mean?
Well, the immense stability of the DNA sequence, protected by the cellular repair machinery, preserves successful lineages. It ensures that complex adaptations aren't just lost to immediate thermal degradation.
Okay, that makes sense.
But the stochastic mutations create slightly altered versions of the code script.
And then natural selection acts as the physical filter.
Exactly.
The variations that happen to increase the organism's ability to capture free energy and reproduce in an unpredictable environment, those are preserved.
Yes.
Life is essentially this high-wire act, balancing the physics of reliability with the biological necessity of randomness.
It's a very delicate balance.
So pulling all of this together, we've established that Schrödinger didn't predict the double helix.
No, he didn't.
His crystal analogy ignores the dynamic nature of DNA. His code script concept misses the distributed reality of cellular heredity. And his negative entropy phrasing is thermodynamically sloppy compared to modern non-equilibrium physics.
When you lay it out like that, it does sound like a comprehensive list of scientific shortcomings.
Right. It kind of does. So why are we still dissecting this book 8 decades later? What is its actual historical value?
Its value really lies in its profound historical influence on the sociology and the trajectory of science. You have to evaluate the book within its context.
Okay.
Before Schrödinger, genetics was largely a phenomenological science. He bred fruit flies and counted the eye colors.
Right. It was observational.
Exactly. What Schrödinger did was rigorously reframe heredity as a fundamental problem of molecular physics.
He asked the physics community a direct question. How can a physical sequence of atoms store information and resist thermal noise long enough to build an organism?
Exactly. By providing a theoretical framework and establishing a set of physical boundary conditions, he made biology intellectually irresistible to a whole generation of physical scientists.
He dared them to figure it out.
He essentially gave physicists permission to invade biology, convinced that the mysteries of life could be decoded using the hard laws of chemistry and thermodynamics. It's like he didn't build the internal combustion engine, but he mathematically proved to a room full of engineers that building a car was theoretically possible, which launched the entire industry.
That is a very accurate way to look at it. However, we must continually emphasize that it was the grueling, subsequent experimental traditions, phage genetics, X-ray crystallography, biochemistry.
Right.
They actually discovered the mechanisms. Schrödinger's theories inspired the workers, but the lab workers built molecular biology.
Which brings us to our final misconception. Misconception number 5.
The reductionist trap.
Yes, the reductionist trap.
Yeah.
Because if we can ultimately explain the material stability of life with quantum chemistry and the energetic maintenance of life with non-equilibrium thermodynamics and the replication of life with molecular biology,
Yeah.
doesn't that render biology obsolete? Isn't biology just applied physics?
That is an incredibly common fallacy,
Right.
and the scientific correction is an absolute no.
Why not?
Because a purely physical explanation does not remove the need for biological and evolutionary levels of explanation. Because physics only provides the syntax, not the semantics.
Exactly.
Physics dictates the constraints. It tells us exactly how much free energy is released by ATP and why a hydrogen bond is stable. But thermodynamics cannot explain why a particular sequence of DNA exists in a population of moths in industrial England, while a completely different sequence exists in the countryside.
Precisely. Physics explains what is materially and energetically possible, but natural selection, ecology, and developmental biology explain the actual historical trajectories, the patterns, and the functions we observe in the real world.
Right.
To understand life, you need the physical mechanics of the how, but you absolutely require the evolutionary context of the why.
They need each other.
They are complementary, irreducible levels of explanation. You cannot understand the plot of a novel merely by analyzing the chemical properties of the ink on the page.
That's a perfect analogy. The ink allows the information to exist physically, but the environment and evolution actually wrote the story. And the story is still expanding. Because the questions Schrödinger codified in 1943 are actually driving the most aggressive frontiers of modern science right now.
Absolutely they are. I mean, in synthetic biology today, researchers are physically writing massive synthetic genomes from scratch and inserting them into nucleated cells just to see if they boot up.
Right.
They're testing exactly where the line is between the informational sequence and the necessary cellular hardware.
And in Origins of Life research, scientists are debating whether life began with an RNA world, like a molecule that could act as both an informational aperiodic solid and a chemical catalyst.
Right.
Or if it began with the metabolism-first approach, focusing entirely on those thermodynamic networks of free energy before genetics even existed.
And when we look outward to astrobiology, Schrödinger's theoretical baseline is still our best guide.
Really? How so?
Well, if we send a probe to the subsurface oceans of Enceladus, we cannot assume alien life will use DNA.
Right. It could be entirely doing chemistry.
Exactly. But we can assume it must adhere to Schrödinger's thermodynamic and informational requirements.
Which are?
It must be an open system, maintaining local order far from equilibrium by consuming free energy. And it must possess some form of stable, aperiodic structural memory to pass on its physical adaptations.
Okay, let's synthesize everything we've unpacked here today.
Yeah.
We started this deep dive with a central question. Can the stability, inheritance, organization, and apparent order of living systems be explained through physics and chemistry?
And the integrated evidence-based answer is yes, but conditionally.
Conditionally.
Right. Physics and chemistry flawlessly explain the material stability of molecules like DNA. They dictate the energetic constraints and the non-equilibrium thermodynamic flows that make cellular life possible.
Okay.
But that physical foundation is only half the picture.
Right.
Molecular genetics, cell biology, and evolutionary theory are absolutely required to explain how that physical information is interpreted by an organized cellular system,
Yeah.
how it drives complex developmental pathways, and how it survives and adapts over billions of years. Physics builds the stage and sets the lighting, but biological evolution directs the play.
This production is an independent COSMORA HQ book overview. It is not an audiobook, narration, adaptation, abridgment, reconstruction, authorized edition, or substitute for lawful access to the original work. To close out this deep dive, we want to leave you with a final thought to mull over based on the realities of the physical cell. Think carefully about this. Which is more essential to understanding life, stored hereditary information, or the organized cellular system that interprets and maintains it?